Hello, everyone. Welcome back. Um, we are now, this week, we're going to talk about biochemistry. And biochemistry is the study of the molecules that make up living things. Um, and if you look at the slide that's up there now, you'll see that there's 6 major groups of biochemicals that make up most of our bodies. Not all, but most of them. Water is by far the most. About 70% of our body is water. Um, Proteins, carbohydrates, lipids or fatty substances mostly, nucleic acids or DNA and RNA, and then there's ATP. ATP we'll talk about next week, but it's basically our energy source. So if you look at what we're going to do this week, we're going to talk about water, proteins, and carbohydrates. And we'll start with water. Water is a very common compound. Obviously, it's everywhere. Comes out of our faucets, it's in the oceans, it's everywhere you look, it's in the rain. Water is a molecule that, let me get back to it, that looks like, kind of looks like Mickey Mouse. It has oxygen and hydrogens, H2O. The 2 stands for the 2 hydrogens. So there's 2 hydrogen atoms bonded to 1 oxygen atom. If you look at the shape of the molecule, you'll see it does have a sort of lopsided shape. There's an oxygen atom in the center with 8 protons and the nucleus, 8 neutrons. The protons have a positive charge. That's very important. The electrons are flying around the outside of that almost at the speed of light. And you'll notice that the 2 hydrogens above, the +1 positive side, they're sharing electrons with the outer energy level or orbital of the oxygen. And the important thing about that is that that creates a bond between the hydrogen and the oxygen, and a very strong bond, in fact, and they call that a covalent bond. It's very difficult to separate the hydrogens from the oxygen of water. But because those 2 hydrogens are up there on the top, like the 2 Mickey Mouse ears, they're actually 104 degrees apart. They have positive nuclei, +1. Those are protons. There's nothing like that at the bottom of the molecule. The bottom of the molecule is just negative electrons flying by. Notice then, because of that, the top of this molecule here in the diagram is positive compared to the bottom of it, which is negative. When you have a molecule like that that has a positive side, negative side, it kind of acts like a magnet. It's called a polar molecule. Polarity is the situation where you have a molecule that has a plus side on one side and a minus side on the other. Now we know that opposite charged molecules or anything actually attract each other. If you have a magnet, the positive end of the magnet would attract the negative end of another magnet, and they'd stick together almost magically, like without even any glue. Opposite charges attract. So if you have another water molecule like the one at the bottom of the diagram, notice that its positive side, the 2 ears of the Mickey Mouse shape, are attracted to the negative side of the molecule above it. And that little bond that's formed between them, sounds like a weak magnetic bond, that bond is called a hydrogen bond. It's not as strong as a covalent bond that's holding the hydrogen and oxygen together, but it's still enough to keep the water molecules stuck together between the temperatures at which it boils and at which it freezes. That makes water at the temperatures between Fahrenheit at 32 degrees and 212 degrees, or in Celsius 0 and 100. It makes water in between those temperatures and makes it a liquid, not a gas. If that hydrogen bond wasn't there, the water molecule would be free to just fly around the air by itself, and it wouldn't attach to any other molecules, therefore act as a gas like the gases in the air today. It wouldn't even be wet. But the molecules that are the water molecule attracting other water molecules make a very large lump of water molecules, like a giant lump of magnets, and that creates what we know as a liquid. So water is a liquid at this temperature, at the temperature at which we live in, because of its polarity and because of the shape of the Mickey Mouse head that it has, the plus side and the minus side. Those hydrogen bonds have a lot to do with the water's properties. So let's take a look at that. In the next slide, it says that such properties are all due to the polarity of water, the plus and minus side, and that's due again to the shape of the molecule. It's, as I said, it's a liquid between 0 and 100 degrees Celsius. In Fahrenheit, that would be 32 and 212. However, because of the polarity, because the molecules are kind of stuck together magnetically, it's tough to get them to move faster. And a very important thing about this is that the difference between heat and temperature. Heat is the amount of energy that's in the substance. The temperature is how fast the molecules are moving. So heat is usually measured in calories. Temperature is measured in degrees, either Celsius or Fahrenheit. But because water molecules are pretty well stuck together, it takes a lot of energy to raise the temperature of water. You got to add a lot of heat to it to get the molecules to start moving faster, because they're kind of pretty well stuck to each other. If you want them to get cooler, you have to take a lot of energy away. So it's tough to change the temperature of water, and that's why it takes so long. For example, the macaroni water on your stove to boil. You have a big pot of water up there. You're hungry. You want your macaroni, and you always wonder, when the heck is this going to boil? Well, you have a fire under it, which is many hundreds of degrees of heat, on there for many minutes, and yet the water is not boiling. You have to wait a pretty long time, because all that heat has to be absorbed by the water to get the molecules to move fast enough to get them to be above the boiling point. If you were going to put the same amount of oil, like olive oil or something, in the pot and put the same amount of heat under it, it would get to the boiling point of water, 100 degrees Celsius, about almost twice as fast as the water would. It would heat up much faster because the molecules are easier to move around. It takes less energy to get the molecules of oil to move faster than it does to take the molecules of water to move faster. Now, why is that important to us? It's important because most of our body is water, 70%. So because most of our body is water, it takes a lot of heat to raise the temperature from 88 to 99 to 100 to 101. And it takes a lot of heat. You have to lose a lot of heat to get it down to 97, 96, 95. And because it takes so much heat to either raise or lower our temperature, it's easier to maintain the temperature at 98. If our liquid in our body was made of oil, we would be changing our temperature like crazy every time we go into a hot or cold environment. But because it's water, it resists temperature change, and therefore we can control our temperature a little bit better at 98, that nice homeostasis. Remember, the Earth is also mostly water on the surface, and so when the sun, beats down on the Earth every day, it heats it up. But because the water in the ocean absorbs that heat so well and doesn't change its temperature that much, it moderates the temperature of the entire planet. If we were on the moon, or we didn't have any water on the Earth, during the day it would be over 200 degrees or more, and at night it would be something like minus 200. So obviously we wouldn't be able to live in those conditions. So that means that polarity of water, which causes it to resist temperature change and resist moving faster, because they're stuck to each other pretty well, keeps our whole planet as well as our body able to maintain life. Also, because of the polarity, water can dissolve a lot of things very well. So a lot of the chemicals in our body obviously wouldn't do their chemistry unless they were dissolved in water, just like in a chemistry class. And so, we have to get this stuff dissolved. And so 90% plus of our cells is basically water, and all the biochemistry that happens inside of those cells is dissolved in that water. And if water wasn't a good solvent, that wouldn't happen, and the chemistry wouldn't happen, and we wouldn't be here. All your cells, you have trillions and trillions of cells with a T. They exist because they have membranes around them. You have to have a boundary between the inside and the outside of the cell. That's the cell membrane. But the cell membranes are actually held together by the little magnets that are on both sides of them. The water on the outside of the membrane and the water on the inside of the membrane is actually working together as little zillions of little magnets to hold that membrane together magnetically. We'll talk about that more when we talk about cells later on in the course. Plants, they can get pretty big. You have a redwood tree that could have a leaf, 200-300 feet in the air. And if you go up there and take that leaf off and cut it in half, you'll see it's wet inside. There's water that got into that leaf. And somehow that water got all the way up that tree from the ground. And one of the things it uses for that, it's not the only thing, but one of the things it uses to get the water uphill like that is the polarity of the water. The magnetic properties of water causes the water molecules to attract to the sides of those little pipes inside the wood, and it causes to creep up the plant. So without the polarity, water would not go up trees very well, or even our food, plants like wheat and corn, and we wouldn't have anything to eat. And even if you only eat meat, obviously a cow is just modified grass, and so you'd be unable to even have any meat. So because the water is polar, we have the plant kingdom, and because we have the plant kingdom, we have the animal kingdom. Also, and last but not least, is that water expands when it freezes, which is very unusual. Most molecules will, when they freeze, they become more dense. They contract when they freeze, and they would sink in the liquid they're made of. Benzene, if you take some benzene or paint thinner or something like that and you freeze it, the frozen molecules would sink straight down to the bottom. But if it's water, water expands when it freezes because of the shape of the molecules. And when it does, it becomes a little bit less dense than the liquid water it came from. So it floats. And because water floats, we know that from having a drink, and ice always just floats on the surface of the glass of water on the top. But because it floats, when something freezes in the winter, like a pond or a lake, or even some saltwater environments or streams, the ice stays on the surface. And that allows the life underneath that to stay alive. If that didn't happen, the algae would never have evolved into the plant kingdom. And we wouldn't have any plants, and we wouldn't have any animals. So the little thing like that ice floats, allowed for life to exist on the planet. Another little slide here about water coming up. It's a little one that has to do with your body. It says here that what does water do for you? Well, obviously you wouldn't have any saliva without liquid water. It keeps your membranes moist, which is absolutely necessary to keep you alive. It allows your body cells to grow, reproduce, and survive because your body cells are mostly water, and the chemicals dissolved in them make the life possible. It flushes your waste, mainly in the urine, lubricates your joints. The major component of most body parts is water. You need it in your brain to manufacture hormones and neurotransmitters. Your brain would not work with liquid water inside of it. Regulate your body temperature, like we spoke about. Acts as a shock absorber in your brain and spinal cord, because if there's water there, your brain is kind of going to resist having a concussion because there's water to act as an absorber for you if you get hit with something. It converts food into components needed for survival. In other words, you couldn't digest your food without the water in your stomach and the water in your intestines. And obviously it helps deliver oxygen all over your body because the oxygen has to be dissolved in your blood. And the blood is, as you know, mostly water. So yeah, water is kind of an important one. Now we're going to switch over to carbohydrates. Now carbohydrates are molecules where the name carbohydrate actually tells you what it is. Carbo means carbon, hyd means hydrogen, and ATE many times in chemistry means something ends in oxygen. So carbohydrate is really telling you carbon, hydrogen, oxygen. Carbohydrates are made of those three atoms. Carbohydrates are made of carbon atoms, hydrogen atoms, and oxygen atoms. Now, if you look at the formulas for carbohydrate, which tell you how many carbons, how many hydrogens, how many oxygens, normally, almost always, there's twice as many hydrogen atoms as there are oxygen atoms in the molecule. Kind of like the same ratio as water, 2 to 1, 2 hydrogens for every oxygen. You'll see a couple of examples of that in the next slide. However, there's three types of carbohydrates that we have to deal with. And they have fancy names, but they're not really that difficult. Mono means one, di means two, and poly means many. If you keep that in mind, it'll make things easier for you. Saccharide is the term that means sweet. So monosaccharide is a simple sugar. They have from 3 to 7 carbons in them. There's many different kinds of them. And they are the monomers. And what we mean by a monomer is something that you can link together to form bigger molecules. For example, if you have a chain made of links, each link in the chain is a monomer. Poly, the prefix that means many, if you had the chain, the whole chain would be a polymer. Many monomers stuck together. So in biochemistry, when you're talking about carbohydrates, when you have the bigger ones, they are usually made of monosaccharides, little simple sugars, joined together just like the links of a chain. The simple sugars would be the monomers, and if there's 2 of them linked together, there would be a disaccharide. If there are many linked together, like 1,000 of them or even more, you'd have what they call a polysaccharide. And we'll talk about that as we go along here now. Now it says dehydration synthesis. Dehydration synthesis is just a simple process that the molecules use to join together. It's the way they link. So obviously if you have 2 links of a chain, somehow you have to join them together. In biochemistry, dehydration synthesis is the joining process. And we're going to take a look now to see how that actually works as we go along here. Now, in the next slide, you see a couple of examples of monosaccharides. Now, you're never going to have to draw these or memorize their structures or anything like that, but just be able to recognize their names at least. Fructose, glucose, and galactose. These are the three most common monosaccharides that we deal with in our bodies. Fructose has 6 carbons, 12 hydrogens, and 6 oxygens. Remember, a carbohydrate is made of 3 kinds of atoms, carbons, hydrogens, and oxygens, usually with twice as many H's as O's, and that's what you got here. 12 H's and 6 O's. Now, if you look at the diagram or the molecular structure of fructose, you can see that you can see the 6 carbons, and if you counted the H, there'd be 12 of them, and 6 oxygens. The lines indicate those covalent bonds, where the atoms' electrons are being shared and therefore linking atoms together. So each dash is a bond. It's a what they call a covalent, very strong covalent bond between the atoms, joining them up and linking them together. Now, an important point now, that's a chemistry point, but you don't have to understand why it is because we're not a chemistry class, but just take my word for it. Carbon's magic number is 4. Carbon always makes 4 bonds with things. Oxygen's magic number is 2. Oxygen always makes 2 bonds. And hydrogen's magic number is 1. So it always makes 1. So if you look at the fructose, for example, you'll see each C there. Now that you know that, each C has 4 bonds connected to it. Each O has 2. The OH is you have to follow to produce these molecules. 4 for carbon, 2 for oxygen, and 1 for hydrogen. However, you can put them together in different configurations as long as you follow that rule. If you look at glucose, for example, there's still 6 carbons and 12 hydrogens and 6 oxygens, and each carbon has 4 bonds, and each oxygen has 2, and each hydrogen has 1, but it's put together in a different shape. Therefore, glucose is not fructose, even though it has the same formula. It has the same number of C's, H's, and O's, but it's not the same substance, and it has different chemical properties. Then if you look at galactose, it's only one little change. If you look at the carbon on the left side, you'll see the OH is on the top and the H is on the bottom of that. Otherwise, it's the same as glucose, but it's still, because it's the configuration is different, it's still a different molecule. And it is also C6H12O6. Now, in chemistry, if two molecules have the same formula, but they're put together differently, like these three are, they all have the same formula, C6H12O6. That's the molecular formula. But they are not the same substances because of their structure. Those are called isomers. So you say glucose, fructose, and galactose are isomers of each other. They're very similar in structure. They would all taste sweet on your tongue, and they are, but they're not the same substances because their structures are different. These guys are mostly used for energy. When we burn them up in our cells for our energy, we eat this, sugary things for energy. But you can link these together. You can take a fructose and link it to a glucose. You can take a fructose and link it to a galactose. You can galactose to a glucose. You can link them together. And when you do, you can make larger carbohydrates. So let's take a look at one of them. These are disaccharides now. A disaccharide are two monosaccharides joined together. And we said a little while ago that dehydration synthesis was the process of linkage, how they link together. I'm going to show you now how that works. If you look at the glucose molecule that you have there, it's the same one you had in the slide before, and the fructose molecule. You see, I kind of pushed them closer together, and I circled the H and the OH, the H on the glucose and the OH on the fructose. And I put a circle around them. There are enzymes in plants. Humans don't do this process. We have no reason to link glucose to fructose. But plants do, like sugar cane and sugar beets, they do that. They have an enzyme. And enzymes are proteins that act as chemical catalysts. They make things happen that normally wouldn't happen fast enough to matter. They speed up things, millions of times faster than they normally would happen. And what this enzyme inside the plant would do is it'll rip that H off the glucose and the OH off the fructose. Now H and O will immediately, when they're broken off their molecules, work together to form a water molecule, H2O. Hence the term dehydration. You're taking out the components of water and creating water molecule out of it. You're dehydrating, you're taking water out. But now remember, the O's can make 2 bonds, and the C's are supposed to make 4 bonds. And so if you take that H off of the O and the OH off of the C to make water out of the H2O, the O needs to make another bond, and the C needs to make another bond. So what they're going to do instantly is they're going to join together. And if you look underneath, you'll see a new bond forming between the O and the C. That process that you see there is dehydration synthesis. Dehydration because you're taking out the enzyme, takes out the components of water. This would not happen without the enzyme being present. The synthesis part means you're building something, and you're building a bond between the glucose and fructose. Now, it turns out that when you link glucose and fructose together like this, you create a disaccharide. Remember, di means 2. And this particular saccharide, glucose and fructose, that's sucrose. That's the exact stuff that you buy in the grocery store. That's sugar. That's what you would buy in the grocery store and use in your kitchen as sugar. It's sucrose. It's a disaccharide made of glucose and fructose linked together. Now, when you put that in your mouth and swallow it, either in cake or in coffee or whatever it is you use it in, you have an enzyme, a different enzyme now, in your stomach and in your intestines. That enzyme will break the bond between the O and the C and put the H back on the O and the OH back on the C, and that will recreate glucose and fructose. And that's how you digest the disaccharide. You're turning it into 2 smaller molecules, glucose and fructose, by breaking the bond. And that process is called hydrolysis or hydrolysis because you are breaking the bond. Lysis means to split. You're breaking it, and you're adding the water components back in. And that makes you have a glucose and a fructose. Now, when those molecules are that small, they can go across the walls of your intestines. It turns out that sucrose itself is twice the size because it's a disaccharide. And that, if you got that to go all the way into your large intestine without digesting it, without breaking that bond, without doing hydrolysis on it, it would not be able to get into your blood, and your poop would basically be sweet. And so basically that is how you digest your sugars. You do a process called hydrolysis. You turn it back into glucose and fructose. The monosaccharides are small enough to get into your blood, go around your body, and feed you. Now, if you look at the next slide here, you'll see a couple of disaccharides that you should know about. One of them is maltose. A dimer means two linked together, two monos linked together. Maltose is glucose linked to another glucose. And maltose is a molecule that you find in beer. You can get a malted milk at a Newport Creamery or something like that, and they'll put that maltose in there for you. It is commonly used in recipes and things like that. You notice that almost all these carbohydrate names end in OSE. That's a dead giveaway that something's a carbohydrate. Some don't. You're going to see some carbohydrates that don't end in OSE, but almost all of them do. Lactose is a very important one. Lactose is glucose and galactose joined together. Remember, we saw galactose as one of the monosaccharides a little while ago. When you join glucose and galactose, you create lactose, and lactose is the disaccharide that's found in milk. Now, remember, when we take disaccharides as food, we have to break them down. We have to do hydrolysis on that bond. We got to break them back into monosaccharides, into glucose and galactose. We need an enzyme to do that. And the enzyme's name would be lactase. ASE ending is an enzyme ending. Maltase would be the enzyme you use to digest maltose. Lactase would be the enzyme you use to digest lactose. Sucrase would be the enzyme to digest sucrose. But some of us have lost the ability to create lactase. And if we can't make that enzyme as we get older in life, you can't digest the sugar in milk. And it stays as lactose or disaccharide lactose. That lactose will end up in the intestine, and what it'll basically do is feed a lot of the bacteria in there, and they'll grow into larger numbers than they should be, because they have all that nice lactose to eat that you're supposed to have, but you didn't do anything with it because you didn't have the lactase enzyme. And so your bacteria will produce a lot of gas and a lot of distress, and you'll have what they call lactose intolerance. That's people who have trouble with milk or any other dairy product that has lactose in it. Sucrose, obviously, as you know, is the sugar that you use in baking. It's the sugar you buy at the grocery store. And you need sucrase to digest that, but I never heard of anyone who can't make that enzyme. The lactase is the one that many people lack, and they can't eat cheese, they can't eat yogurt, they can't drink milk, they can't have cream, all the things that would be rich in lactose, or else they would have intestinal distress because the lactose won't be digested and it will stay in their body all the way to their large intestine and cause problems. Polysaccharides are the last group. Now, polysaccharides are just hundreds or even thousands of monosaccharides linked together by that dehydration synthesis process. Amylose, commonly known as starch, is made of thousands and thousands of glucose molecules joined together. And it's made only by plants, because plants make glucose as their food. That's the sugar they make in photosynthesis, and when they get hit by the light of the sun, and they produce their glucose. But in order to save the glucoses in the leaves and in the stems, and even in the fruits and even in the roots, sometimes the plants link glucoses together. It's kind of like putting money in the bank, and to create this enormous starch molecule. Thousands of glucoses joined together in a big long chain. And because of that, the glucose can't leak out of the cells, and the plant can use it whenever they need to. It's like having reserves. But we can obviously eat the starch and use it for ourselves. Cellulose is also many glucoses joined together, but the bond between the glucoses are between different carbons and different oxygens. The bond just forms in a different location between the molecules. And because of that, the molecules are different shape. And because it's a different shape, the amylase enzyme that we have that digests starch for us, and we can create glucose from it and use it as our food source. We don't make cellulase. Humans don't make this enzyme. So if you eat cellulose, you cannot digest it. It just stays as cellulose, a long polysaccharide molecule that will never get into your bloodstream. Because it's undigestible by you and most of your bacteria as well, it becomes the fiber that most of your waste is made out of when you poop. Cellulose is found in the cell walls of plants. Plant cells have a membrane just like we do, but they also have a wall around the membrane for more support. And we'll talk about that later on when we talk about cells. But those walls are made of cellulose. And every single plant cell has a cell wall. So every single time you eat plants, you're eating fiber. And you're not able to digest it, it would just go through your intestine. And that's healthy. You want to make sure you eat enough fiber to keep your intestine working right and not become things like diverticulosis, where it works on too small pieces of waste and kind of has to stretch and strain to move it along, and it gets a little bit out of shape. You want to make sure enough fiber in your intestine to keep it healthy. And cellulose is a major fiber source. Glycogen is the way we link our glucoses together. If we have more glucose than we need in our blood because we're eating a lot of carbohydrates, you don't want to keep that in your blood because you want to keep the glucose level at a certain, in between certain numbers, homeostasis. You don't want to get too high. So what you can do is you can link glucoses together to form what they call animal starch or glycogen. And you store that actually in your liver. And so you have about 2 or 3 days worth of glycogen in your liver in case you didn't eat in the next 2 or 3 days. That would be able to provide you with enough glucose, if you break it down, for your blood sugar to stay, within limits. If you have glycogen, it's very, very important that you look at how diabetes works. If you have a problem with glycogen and glucose, it's the diabetes problem, and you'd probably end up with too much glucose in your bloodstream, and then that could cause problems for almost all your organs. And we could talk about that later again when we talk about our cells. But glycogen is the way humans and other animals store their glucose. They link them together in big long chains and create this long molecule we store in our livers. And then we can break it down into glucose little by little as we need it. Chitin, kind of unimportant to us, but it kind of is, because chitin is also undigestible, so we can use it as a fiber source for our intestines. It's made of amino sugars, which is something we'll talk about when we talk about our proteins later. But they're found in the cell walls of fungi. Fungi would be like mushrooms and things like that. If you eat them, you're eating fiber because the cell walls around the mushroom cells are made of chitin, and that is not digestible. The outside of insects, their outside skeleton, they have no bones. Their skeletons on the outside, they're like little tanks on the outside. They're hard on the outside, soft on the inside. But those outside shells around the insect, they're made of chitin too. And there are some cultures in the world that actually do eat insects, and they're getting a good amount of fiber as well as low cholesterol, high protein, and low fat food for themselves. But we know in this country we try to frown on eating insects. But we do like lobsters and crab and shrimp, and they're basically in the same phylum as insects. But as long as they come from the sea, I guess it's fine. But a lot of people eat grasshoppers and crickets and things like that, especially in Asia, and it is a healthy food source. Now, quickly going over to proteins to finish up our lecture today. Proteins are polymers also. They're made of chains of amino acids. Now, instead of being chains of glucoses like carbohydrates are, they're chains of something called amino acids. Amino acids, I'll show you one soon, but amino acids are small little molecules. They have carbon and hydrogen and oxygen also, but they also have a nitrogen atom in them. That's one of the reasons why you have to have nitrogen fertilizer on your lawn, because plants obviously don't eat and they can't get amino acids from their food like we can. And so they have to make their own amino acids, and in order to do that, they need nitrogen. Nitrogen is one of the elements in amino acids. So without nitrogen in the soil, the plants would have trouble with their proteins, and of course that's serious. Now, when amino acids join together, they do it by dehydration synthesis, same thing. Enzymes take OHs off of one side and H's off the other, and the HHO becomes water, and the broken bonds join together. Same story. When they do that, they form a long chain of amino acids, sometimes up to 1,000 amino acids long. Some of them can be shorter, some longer. Depends on the protein. But eventually that chain will fold up. And when it folds up, it becomes a shape. The molecule becomes a certain shape. And it's the shape of the molecule that gives the protein its ability to do its job. It's kind of like the shape of a key. The key shape is everything about it. If it's the right shape, it'll open the key, open the lock, even if it's made out of wood. But if you change the shape even just a little bit, it probably won't work anymore. And that's true also for proteins. The amino acid chain has to fold up into a shape, and the shape then does a job in the cell. Now, what's the difference between one protein and another one? Well, it turns out that there's 20 different kinds of amino acids. Now, this is not a biochemistry class, so we don't have to know the names of all 20 amino acids, but there are 20, and you should know that number at least. And each one of the 20 amino acids are different from each other slightly. And so if you have 20 of them, they can join together in different combinations. Just like you have 26 letters in the alphabet, and they can join together to form words. And you can imagine how many words there are in the English, French, Spanish, Italian, German dictionaries. And they're all basically made of the same 26 letters. Joining things together like that is very powerful. So if you have 20 kinds of amino acids, you can join them together in different combinations, different orders and different numbers of them to create all the different proteins in the world. The only difference between, let's say, the protein in your hair and the protein in a piece of cheese is the number and order of the amino acids in the chains. It's the same 20 amino acids in both things. All proteins basically on Earth are made of the same 20. They're just different because they're in different order, just like you spell a word differently, or different number of them. And now I'll show you what one looks like. Here's the next slide. Remember, the carbon makes 4 bonds, oxygen makes 2, and hydrogen makes 1. And you'll notice that there's an N there. Nitrogen's magic number is 3. So when nitrogen makes a bond, it always makes 3 of them. And so you see there that there's 3 lines going to the nitrogen. Now that N with the 2 H's on it, that is known in chemistry as an amino group of atoms, NH. It's very common. It's found in many, many different things. And it's always called an amino group. On the other side, you have a C with 2 bonds to an O, so that's the oxygen's double bond, and a C to another, an O, and then an H. You notice, though, that H has a plus sign on it. When atoms are charged like that, they have an electrical charge to them. They call them ions, I-O-N-S, ions. So that's not a hydrogen atom on the other end there. That's called a hydrogen ion. And any chemical that has hydrogen ions in it, and that would increase the hydrogen ion concentration in a solution if you added it to it, that's called an acid in chemistry. So you notice this molecule has an amino group on one side and an H plus or an acid ion on the other side. Therefore, its name is amino acid. The COO, that's just called a carboxyl group because it's just carbon and oxygen. Now, it says down there are 20 different amino acids. And they're only different in one thing, what that R stands for. That R can stand for 20 different chemical groups. If this was biochemistry, you'd have to look at a chemical group up there and identify which amino acid it is. We, thank God, don't have to do that. All you have to know is that the only difference between one amino acid and another one is the same everywhere except for that R. There could be many different things. It can stand for H, a C with 3 H's on it. It could be a ring-shaped structure of atoms. There's 20 different R's. You don't have to know what they are, just that they exist as 20 different ones. And they make one amino acid different from the rest. If you go to the next slide, you'll see how proteins are joined together. Now, this particular amino acid here, this is glycine. Glycine has an R group on top here, which is just a hydrogen atom. It's the simplest of all of the amino acids. The second one here is alanine. Okay, so this one's glycine here, and this one's alanine. Notice it's all the same on the bottom here. The only difference is that on the top of the middle carbon, there is a C with 3 H's. So that makes it a different amino acid. It's a different R group. But you'll notice there's no difference here. In order to create this, you have enzymes in your cells. None of this stuff works without enzymes. We'll talk about that more later on. But it'll take the OH off of the glycine and the H off of the nitrogen of the alanine, and they will instantly form water, which is down here, see? Will form the water molecule. And because the O needs to make 2 bonds and the N needs to make 3, the O is going to rejoin with the N instantly to make that new bond. And you'll see it here between the C and, the C will join to the nitrogen to make a new bond because the O would be taken off, you're right. And the C needs 4, so that C is going to remake its 4 by lining up with that nitrogen. And you end up with a bond between the C and the N. C is back to 4, N is back to 3, so everybody's happy. And that bond between amino acids is just a regular covalent bond, like electrons being shared, holding everything together. But because it's between amino acids, and it's going to be part of a protein, and peptide is a word that has to do with proteins, they call that a peptide bond. So now we have 2 amino acids joined together. Now you can imagine a third amino acid doing the same thing here, and then the 4th one, and 5th one, and the 6th one, and eventually you can build a whole protein by linking these amino acids together. And remember, the only difference between one protein and another protein is what order the different 20 amino acids are in and how many of them there are in the chain. So here is a chain of amino acids, and you see step by step, it's going to fold up into a shape. And this is the protein shape. And you see right there's a little notch. That's probably the important part of the protein. That's like the part of the key that turns the lock. That's going to link onto something and make something happen in the cell properly. If that notch was a different shape because there's one amino acid missing in the protein or the wrong one is put in and it folds up a little differently, the protein might not work at all. And a lot of genetic disorders are caused by that. They're caused by proteins that have incorrect amino acids in their chain and not the right one in the right place. And that causes the problem. Now, what's some of the functions that proteins have? This is our last slide for today now. Most of your body, except for water, is made of protein. Your skin is mostly protein. Your muscles are mostly protein. Your eyes are mostly protein. All your organs are mostly protein. Even your blood vessels are mostly protein. Proteins are basically what you're made of. Your fingernails and toenails and hair, all those things are proteins. So they're structural. Proteins. Different proteins produce different parts of your body, and other parts of it. Some proteins can transport things like little taxi cabs. Some proteins can grab onto oxygen, for example, in your lungs and take it to the cells of your body. That protein would be called hemoglobin. It has a certain chain of amino acids in a certain sequence, and if you do them in the order, you create hemoglobin. And hemoglobin is a magnet for oxygen. It's in your blood. It makes your blood red. And what it does is it sucks up the oxygen from your lungs and takes it to your cells. just like a taxi cab would do. Because you have hemoglobin in your blood, you can take about 30 times more oxygen around your blood at a time than you could if the oxygen just dissolved in your blood. So yeah, hemoglobin is very, very important as a transport protein. Some of your hormones, not all of them, but some of your hormones like insulin and growth hormone, those are proteins. And they're very, very powerful. I mean, look what growth hormone does to you. You start out as a little baby, you end up 6 feet tall. All kinds of issues. Insulin would, affect your sugar level in your blood and keep you from getting diabetes. These things are hormones. They're made in glands in your body and are used throughout the whole body to control things. They are very important. And we're going to have a whole class on hormones later on in the year. Contractility means that some proteins can contract with force, kind of like a rubber band when you let it go. And that is a simplified way of kind of describing how muscles work. Muscles only work by contracting their proteins and cause you to move your bones and be able to walk and talk and do all the things you do with muscles. Antibodies are proteins in your blood that protect you from disease. You make antibodies when you get sick to fight the disease, and then you get better. If you couldn't make the antibodies, you probably wouldn't get better of anything. In fact, people who die of HIV infection and get AIDS, they die because the virus that causes that kills the cells that make the antibodies. And so you die of a whole lot of things at once. It's horrible. Enzymes, as we already talked about, are catalysts. They create the chemistry. They allow the chemistry to happen in our cells fast enough. Just about all of the thousands of chemical reactions that happen inside of our cells are controlled by enzymes. If you don't make the enzyme, you don't do the reaction. That's one of the ways that DNA controls you. If DNA wants a certain reaction to happen in a cell, it will make the enzyme, and the enzyme will cause that reaction to happen. And enzymes are by and large all proteins. Very important. You wouldn't even be able to digest your food without the proper enzymes. Cellular identification, that means that on the outside of your cells, you have proteins that act as almost like little signposts. They tell other cells what kind of cell that is. Is it you or not? For example, your immune system has to be able to tell the difference between cells that belong to you and cells that are invaders in your body. They have different ID proteins on the outside, different uniforms. And so your immune system can differentiate between them, leave your cells alone, and kill the bad guys. Every one of your cells have proteins on the outside of them that identify them. Your blood type proteins do that for your blood cells, type A, type B, type AB. Those are identification proteins on the outside of blood cells that tell your immune system that those cells are you. If you get a transfusion of the wrong type, your immune system might actually attack it because it will recognize it as cells that don't belong to you. Receptors, we'll talk about more about them when we talk about muscles and nerves, but there's a lot of times in your body where molecules or cells actually have to communicate with other cells, and there's a little space between the cells. So what the cell does is it makes a chemical, usually a protein of some kind or some other type of chemical, that goes into this space, and there are protein receptors on the other side of the space, and the chemical lands on those receptors, kind of like your finger landing on a doorbell, and makes something in the receiving cell. Those are called neurotransmitters, and that's very, very important. Your brain works through them, your muscles work through them, all your nerves work through them. If you couldn't communicate nerve cell to nerve cell or nerve cell to muscle cell, you wouldn't be able to control your body at all. And that's how things happen when people lose control. If they shake, if they have Parkinson's disease or something like that. A lot of times it's because of these neurotransmitters that are supposed to go across these spaces to have one cell communicate with the other. The receptor is not there, or it doesn't take it right, or something goes wrong with the process. And so the cell doesn't get the message. So receptors are protein molecules that are on the outside of cells that are waiting for molecules called neurotransmitters to attach to them to cause something to happen in the cell. Without the neurotransmitter attaching, a cell, for example, a muscle cell, would not contract properly. Membrane pumps, the last thing. Membrane pumps are proteins that are embedded in your cell membranes that actually use energy to pump molecules, atoms, and ions into or out of the cell. A lot of things can't get through the cell membrane fast enough that the cell needs to either get rid of them or take them in from the outside, and so the cell absolutely pumps them in or pumps them out. In fact, you use a tremendous amount of your energy from your food to power those pumps in all of the trillions of cells to pump things in and out. So you can see that proteins are structural. You're made of them. They transport things around your body for you. They act as hormones, contract like muscles. They make antibodies so you can get better when you're sick. They control all your chemical reactions as enzymes. They tell your body which cells are yours and which cells are not. They are receptors, so one cell can communicate with the next, and they can pump things in and out of the cells using energy. So hence, they're important, very, very important. So that's what we have for this week. This week, we talked about water, carbohydrates, and proteins. Next week, we'll talk about lipids, DNA, RNA, and ATP. Okay, so I'm going to make this video available pretty soon, and you'll be able to watch it and do the homework assignments. See you all next time.